Welcome. Today I want to discuss the basic characters of phytochrome, which is one of the most important photomorphogenetic chromoprotein. At first, we must have an idea that light is crucial for all types of biological activities. It may be photosynthesis in plants and other growth and developmental purposes or activities in plants. So, the light-mediated changes in plant growth and development, which is completely independent from photosynthesis, are commonly known as photomorphogenesis. In this type of activities, light acts as a signal to initiate and regulate the particular process. And in this photomorphogenetic systems, there are two types of light sensing systems which were involved. One is the blue light sensitive system, represented by cryptochrome and phototropin, and the second one is known as the red light sensitive system, regulated by phytochrome. The phytochrome, which I already mentioned that it's a photomorphogenetic chromoprotein, basically a blue pigment with a molecular mass of 125 kilodalton. And actually they found in two interconvertible forms. One is known as the red light absorbing form or the PR form, and the second one is known as the far red light absorbing form or PFR. Now, if we characterize these two forms, the PR form or the red light absorbing form, it absorbs at 666 to 670 nanometer, blue in color, and most importantly, this is actually an inactive form. And this form is synthesized in seedlings. And when it absorbs the red light, it is converted to the PFR form. Now, if we consider the PFR form, it absorbs at a peak of 730 nanometer, green in color, and it is the active form, which is responsible for the photomorphogenetic responses. When the PFR form absorbs far red light, it is converted into the PR form. And most interestingly, this PFR form can be converted into the PR form spontaneously. And this spontaneous conversion in dark period, this is known as dark reversion. dark reversion. Now, phytochrome, if we consider the chemical nature of phytochrome, it's a family of chromoproteins with a small covalently bounded pigment molecules. Phytochrome proteins occur as a dimer, and each of these have a molecular weight of 125 kilodalton. Each polypeptide is covalently attached pigment molecule, and this pigment molecule is known as chromophore. Actually, this is a liner tetrapyrrole, commonly known as phytochromobilin, and we can consider its similarity with the mammalian bile pigments. This is the basic structure of phytochrome. Now, it is interesting to note that phytochromobilin, that is the chromophore part, this is, and already I mentioned that it's a liner tetrapyrrole. And this phytochromobilin is synthesized in the plastids, and its precursor is delta aminolevulinic acid. So here we get the precursor, delta aminolevulinic acid. And together the apoprotein, that is the polypeptide chain, and this chromophore form the holoprotein, or the complete protein part. Now, the assembly of apoprotein with its chromophore is completely automatic or autocatalytic and occurs spontaneously. When the chromophore absorbs light, there is a minute change in the structure or conformation, which causes the conformation of the protein portion to the form that initiate the particular photomorphogenetic responses. Actually, upon absorption of light, the PR form or inactivated form, which already I mentioned, and especially the chromophore part of the PR form, undergoes a cis-trans isomerization of the double bond between the carbon 15 and carbon 16, and the rotation of the carbon 14 and 15 single bond, and this results into the conversion into PFR form. Now, if we consider the phytochrome classes, these are of two types: type 1 phytochrome and type 2 phytochrome. And basically, all types of phytochrome are encoded by PHY. class of gene, PHY. If we consider the type 1 phytochrome, it is completely encoded by the PHYA gene. And if we consider the type 2 type of phytochrome, these are encoded by PHYB, PHYC, PHYD, and PHYE type of genes. Now, PHYA gene and its product, that is the type 1 phytochrome, is abundant in etiolated tissues. Etiolated means dark grown, like etiolated seedlings. And type 2 type of phytochrome is present at low levels in both light grown and dark grown plants, because its genes are constitutively visualized at very minute levels, and the protein is very much stable in the most active, that is the PFR form of the phytochrome. Now we consider the mechanism of action, how phytochrome interacts and provides the photomorphogenetic responses. As we all know that phytochrome possesses a serine or threonine kinase activity, and parts of its structure show a similarity with histidine kinase, similar with bacterial chemotaxis. When activated by the red light or red light exposure, the phytochrome phosphorylates itself, and then it phosphorylates the other proteins in that particular cell. And if we consider the type of the response, either the activated phytochrome migrates to the nucleus, where it interacts with the regulatory mechanisms or proteins, which results into gene transcription. Or in other case, the activated phytochrome completely move towards the cytoplasm, and then after subsequent interaction with other proteins, they migrate into the nucleus to participate in the gene regulatory mechanisms and transcriptional activities. So firstly, in the step 1, on the red light exposure, this is the inactive phytochrome. After red light exposure, it converts into light-mediated autophosphorylation and subsequent activation. Phosphate groups are attached, and it triggers the activational process. Now, this activated phytochrome migrated into the nucleus, and ultimately activation of transcription of light response genes. And finally, the photomorphogenetic responses. So upon light activation, phytochrome converts from inactive to the active form, and then it migrates to the nucleus, and then finally activation of transcriptional machinery. And finally, these activated phytochrome very much involved with various physiological functions like flowering, development of the chloroplast, leaf senescence, the leaf abscission, seed germination, etc. In this case, I must mention the basic understanding for these two terms, senescence and abscission. They are very much correlated. Senescence is synonymous with aging. We use the term senescence basically for plants. And abscission, this actually explains the shedding of plant parts like leaves, branches, fruits, etc. And when a particular plant receives a signal of senescence, and after its completion, it participates in the abscissional process. So in this way, phytochrome take part in various major photomorphogenetic activities of plants. Thank you.